Sorry, we have a lot to cover today. I'm going to try to get through as many of the slides as I can. However, I'm not intending to finish all of the slides if I don't make them through today, so you don't really have to worry if we don't finish all the slides. So I don't know if you're aware of this or not, but at the University of Toronto over the next few days, we actually are really lucky in the sense that we're a big enough university that several award-winning talks are going to be given here at the university. If you have time tomorrow, for example, all day long, you'll be able to hear some Nobel Prize level. They're not Nobel Prize laureates, but they're Gardner Prize winners, and you'll be able to hear them give a talk right here in this room starting at 9 o'clock tomorrow. It goes all the way to 4 o'clock. These are fantastic talks. I really strongly recommend that if you have the opportunity to go and hear about cutting edge research that's being done all over the world, they're coming here. They get to present their research to you. So I'm going to tell you a little bit about some of the other Nobel Prize laureates that we've sort of looked at inadvertently in other aspects. So I'm going to talk to you about two of these individuals, Rita Levi-Montalcini as well as Stanley Cohen. So Dr. Levi-Montalcini actually passed away a couple of years ago. She was one of the biggest female neuroscientists, a mentor, role model, and one of those women in science that actually started this whole campaign of actually making sure that women in science were recognized for their important contributions. And she had the opportunity to work with this biochemist, Stanley Cohen, to help elucidate some of the things that I'm going to be talking to you about today. Okay, so if we're taking a look at what we've been covering so far, so these are some things that I wanted to share with you. I'm also dedicating this lecture to Professor Mike Wiley. He's retired, but he was one of the profs that really influenced my life in the sense that he was the only prof that actually tried to explain things to me. He actually took time out from his schedule to invite me into his office to go through slides and everything else. And he actually told me that you might one day be teaching here at the university, which hadn't entered my mind. And I was actually very grateful to have him as sort of my mentor. He was one of those individuals who also really got me into anatomy as well as embryology. So since that time, I've really been very interested in the whole field of embryology as well as anatomy. So these next few slides are ones that I'd like you to take into context that I've already talked to you about. So when we've been looking at some of these different factors, and we've been looking at things related to depression. And then from depression, we started talking about exercise because exercise has been implicated in being antidepressive. And again, all of the research suggests that this is 100% true. And we've also have looked previously at the way in which different antidepressants work, whether it is through the serotonin and norepinephrine pathway. One of the things that we know that ultimately this leads to a change in gene transcription. Could be in a number of different cells. It could be in cells that are undergoing neurogenesis, etc. But we know that these pathways are incredibly important. A lot of these involve intracellular messaging systems, whether it's cyclic AMP and an increase in protein kinase A. All of these eventually will lead to things that will cause the transcriptional regulators to become activated, and then in turn will cause the neurons to upregulate this factor that we spent a lot of time on, known as BDNF. BDNF, again, is a trophic factor. It causes neurons to stay alive. It also causes neurons to grow, and that's going to be the main emphasis of today's talk. And again, just to link this back to what we had talked about before, we had looked at some of the controversies around exercise. We know that exercise is ultimately good for you. In fact, there was a talk earlier today that really strongly suggested that exercise is something that everyone should be taking part in. So you should be engaging in exercise that leaves you breathless about twice a day. And we know that there are a lot of different factors that go into it. We know that much of these are mediated by these skeletal myokines, and these skeletal myokines are worked into the blood. It may be a factor like irisin, and we spoke last about how irisin is still a very controversial molecule. Whether or not it actually exists, that was one of the controversies that we went through in the last lecture. But we do know that this panel over here, as far as we can tell, on the right, is correct. We know that exercise through the activity of skeletal muscle will increase PGC1 alpha. PGC1 alpha and other factors like ERR alpha will ultimately cause changes in the level of FNDC5 on skeletal muscle. And as we increase the amount of FNDC5, whether that's through the formation of more iRisin or not, will ultimately work on the brain, much like these factors did over here, to cause the transcription and translation of factors like BDNF. And that's why we think exercise is actually beneficial for you in general. And having more BDNF, or brain-derived neurotrophic factor, is incredibly important for us. It's important for you because protecting your brains now at an early age is now becoming more and more evident to be important later in your life as well. So the BDNF is a factor that's important for nerve cell survival, and we spoke very briefly about that. It's also important to differentiate those neurons, so any neurogenesis that ultimately forms differentiated neurons also requires a little push from BDNF. And of course, as I'll talk to you about today, neuronal plasticity, the modification of synapses, also requires BDNF as well. All of these things, taken together as a whole, lead to changes in cognition, learning, as well as in memory. And so the take-home message up until today has been, if you have more BDNF, it's good for you. And I think that all of you would agree, exercise somehow, whether it's through iRisin or through other mechanisms, will ultimately lead to a change in BDNF, and increases in BDNF are good for your brain. You all agree on that? Yeah? So if you are in agreement on that, I'm going to try to change your minds just a little bit. And I'm going to take you through not exactly a controversy, but actually a really important finding. So I'm going to take you through neurotrophic factors because I want you to understand that. And I want you to understand how these growth factors, these neurotrophic growth factors, are important in brain development, especially one molecule that we spent some time on, BDNF. There are a number of different pathways that are involved in BDNF expression and how it is processed within neurons. But I'm going to also tell you that BDNF isn't all that important. It really depends on all of you. It depends on what you were born with genetically. So you might have really good BDNF, or you may not have such great BDNF. I can guarantee you, because you're in university and you've gone through at least 12 to 13 years of schooling, that the vast majority of you, in fact, I would guess that 100% of you actually have one isoform. If I were to genotype everyone sitting here in this room, all of you would have one particular polymorphism of BDNF. And I'm going to try to share with you why that BDNF polymorphism is so important. I'm going to try to finish off the lecture around there. And if I can't make it past that, I'll talk in the next lecture about any slides that I need to cover. One of the first lectures that I had here at the university that actually made sense to me, that actually was really stimulating, that I actually got really invested in, was this whole talk about neurotrophic factors. And one of the experiments that Dr. Wiley was showing us in class was this classic experiment that was done by Victor Hamburger in the 1920s. And it was one of those talks that actually gave me goosebumps. When I was sitting there in the lecture, it was like, wow, that sounds so incredible. I'd want to go out and do a very similar type of experiment. And Victor Hamburger in the 1920s did like the best experiments that he could at the time. And he didn't have a formal lab. He actually did all of this in his basement. And so he was raising these chicks, and these chicks were undergoing what is known as limb bud ablation. So he would take in this long scalpel-like surgical instrument. He would find the limb bud, and he would excise the limb bud. He would surgically remove this limb bud. Now, why is that important? Actually, he had no idea. He just wanted to try doing it and see what the consequences were. But he did know that these limb buds in the 1920s, that they were innervated by or had nervous system input from neurons that originated within the spinal cord. And when he did this, and when he caused this ablation of this limb bud, and he looked at the spinal cord 1 week later, when he looked at the spinal cord with the missing limb, and he took out a spinal cord slice, and he stained for neuronal cell bodies, things like Nissl staining will show you neuronal cell bodies. One of the things that he saw, that within the ventral horn, that the part that actually had the missing limb had very, very few neurons. Now, that was a really important finding. He actually dismissed this finding. He wasn't sure that what he was seeing was actually correct. But he did this experiment several times. He was able to prove several times in different experiments that whenever he did this limb bud ablation, only on the side that had the missing limb would he see that there were neurons missing from the ventral horn, right? And it makes sense that the limb is innervated by motor neurons and sensory neurons, and especially the motor neurons in this missing limb actually had no neurons left over. So there were a reduced number of these sensory and motor neurons in the ventral horn of the spinal cord. And this is, again, Victor Hamburger. So can you guess like what experimental animal he would have used? How could you do these limb bud ablations? So I give you a lab protocol, because this is what we now try to do in third-year labs to get you to think and come up with the experiments yourselves. If I told you to go and prove that a limb bud ablation would actually cause this type of degeneration in the spinal cord, what animal model would you use? Remember, that animal has to survive for 1 week. And of course, you're all shouting or waiting to tell me that it would be the chick, right? So this is a little chick egg. You remove the top of the shell, and you can actually look down into the developing embryo, and you can actually excise the limb at any particular time. You put the shell back on top, it goes back in the incubator, and it develops normally. We couldn't do this, for example, easily in embryonic mice or rats or any other type of animal model. He did another experiment, though, so he got really excited. This seems to be a real effect. He got really excited, and he was wondering, what happens if I actually put extra limbs on this chick embryo? So he was, again, somewhat of a pioneer in thinking a little outside of the box, and he decided that he would add an extra limb to the side that he would normally take it off. So now he's not removing a limb. He's adding an extra limb bud to the area where one limb bud already exists. So on this side, there's an extra limb bud added. He also waits 1 week later, and this is the result that he sees. He sees that on the side that actually has an extra limb bud, he sees many, many more of these sensory and motor neurons on the ventral horn of the spinal cord. Again, a thrilling result. Remove a limb, get less neurons. Add an extra limb bud, you actually get more neurons. And again, compared to the normal. So this gets you to think a little bit. So if you were Victor Hamburger from both of these experiments, but particularly the latter experiment, somehow these different targets, the limb buds, if you remove it or add a limb bud, might help to recruit undifferentiated cells, ones that aren't really neurons yet, to develop into neurons or to cause proliferation. That was his hypothesis. Do you agree with his hypothesis? So you're either, is this hypothesis accurate, inaccurate, or you can't tell? It's a little bit of a trick question, because all of you now in 2017 would be telling me that neurotrophic factor from the limb is actually keeping these neurons alive. He wasn't able, based on those two experiments, actually able to tell you if there's proliferation or if there is differentiation going on. But that was his working hypothesis. It wasn't that they were keeping neurons alive. It was actually that somehow some magic factor in that limb bud, in the muscle, just like we look at muscle and exercise, some magic factor was keeping or differentiating some unknown cell in the spinal cord to differentiate quicker into these neurons or to proliferate those neurons early on. He had no way to know, and he had no way to actually go any further with these experiments. And in the late 1930s, he was actually really lucky, because this is when Rita Levi-Montalcini joins his lab. And they work on this. She's also able to reproduce his results. She stays with him and decides to work more formally with him and the university. And one of the things that they decided was, maybe it's not about some neurogenetic cell, an undifferentiated cell becoming a neuron. Maybe it's these neurons are surviving with some unknown factor. So it's not some unknown target-derived factor that converts undifferentiated cells, but it's keeping those neurons alive. Those neurons need some kind of nourishment that's coming from this source, and if you remove it, that source of nourishment disappears, they die, or if you add more, that means that more of them stay alive longer. So working together from 1942 to 1949, that they were actually able to measure cell death. So they knew that neurons within the spinal cord were actually dying, that they needed factors from muscle, and that without these factors, neurons actually died quicker. So they knew that there was cell death, a lot of cell death in these neurons, and that these factors were being produced by the muscle limb buds themselves. This was actually considered in 1949 one of the foundational experiments within cell biology, not just neuroscience, but in cell biology, that one tissue was able to secrete factors that worked at a distance to keep other cells alive. This was the beginning. Back in 1949, you can actually go and read the original monograph in the Journal of Experimental Zoology in 1949 about their hypothesis that somehow these limb buds actually produce neurotrophins that will keep these different neurons in the spinal cord alive. In other words, this neurotrophic hypothesis says that targets of innervation will secrete very, very small or limiting amounts of survival factors that will allow there to be a balance between the number of neurons that innervate a target tissue and the size of the target tissue itself. There was a lot of evidence after 1949 from other labs that actually suggested this hypothesis seems to be correct. This is a good, solid hypothesis. This was, by the way, originally proposed to be for peripheral neurons. So this was, again, thought to involve neurons outside of the spinal cord. That's where I'll show you this next. But this is also now thought to occur within the CNS as well. Neurons inside the brain and spinal cord directly, without sending out projections to target muscle, are also thought to have neurotrophic factors as well. But they had no idea what this was. This is actually really frustrating. So imagine you were able to discover this magic phenomenon, reproducible by everyone. You know that if you were to take out limb buds, that somehow neurons would die, and if you added more neurons would survive, but you can't find this magic factor. And people wanted to know how to do this. This is also in the 1940s or early 1950s, where there are very, very limited techniques that are available. And so they had to wait. They had to wait for science to actually catch up to where they were able to start thinking about how could we do this based on the current state of technology. And again, a lot of this was brute force biochemistry. So hats off to the biochemists for being able to do these types of experiments. So one of the big factors that Rita Levi-Montalcini decided to do, the original model was to take a limb bud from a developing chick embryo, take it out or add an extra one. That was really cumbersome. She didn't like doing this type of assay. It actually meant that she had to keep incubators around, that she could go back in and look at the eggs themselves, have to take them out and back in the incubator. She wasn't really happy with that. So she developed what is now known as a neurite outgrowth assay. Many of you doing cell culture assays will look for neurites. And these long extensions that are coming out are these different types of neurites. This is not a good example of neurite outgrowth. There are very few neurites going out. And here's an example of a really good neurite outgrowth assay. Here, this tissue has been exposed to a substance where neurites, these long protrusions that you see here, are being projected out. And this is what she had developed. Now, what is this tissue? Any ideas? What is that? So what did they plop into the middle of a cell culture dish and then see if neurites would grow out from that tissue? Any ideas? This is a classic in science. Yes. Cell body of a neuron. Okay, so possibly. In fact, I'm going to tell you that it's not one cell body, it's many cell bodies. Any other ideas? So that's a good start. Anyone else with ideas to help Nancy? So if you're thinking the ciliary ganglion, right? Because that's all you think about all the time. The ciliary ganglion. Of course it's the ciliary ganglion. Do you even know where the ciliary ganglion is? So the ganglia, the group of neurons, in other words, that's a ganglia, the group of neurons that actually control autonomic functions, including things like things in the eye, were the neuron cell bodies that you see here, a collection of them. This is the ganglia. the ciliary ganglia that you see here that control eye changes in the eye, and they decided that this is what we're going to use. If there is going to be not just cell survival, because it has to survive, but we also get neurite outgrowth when we add these different factors, then we know that we found this magic substance. So they start doing that. They literally start grinding up every single tissue within the body. They grind up the muscle, because that's a logical choice. They dump it on here. Do neurites grow out? Okay, great. If we peel back the muscle and we just look at the collagen fibers around the muscle and we grind it up and we dump it on here, does it do the same thing? No, it doesn't. So they do this over and over again to eventually come up with the substance which they called nerve growth factor, or NGF. They didn't really know what NGF was. NGF was purified in 1960. So again, it's about 11 years since they came up with their neurotrophic hypothesis. Science sometimes works incredibly slowly, and over that 11 years, they eventually get what they think is called NGF, or nerve growth factor. And Rita Levi-Montalcini is still unhappy with this, because people are doubting her work and saying that, but you're not 100% sure that other cells or other factors are in there. So again, using some really sophisticated biochemical techniques, in 1969 they were able to show on a protein gel, as well as through other biochemical techniques, that they had only NGF. There was no other substance there. When they did these assays, when they added NGF to it, the things grew out like crazy. Their neurite assay worked incredibly well. So this was where this NGF, nerve growth factor, and neurite outgrowth assay became really important. And again, many, many years later, in 1986, they were awarded the Nobel Prize for identifying the first nerve growth factor, or the first neurotrophin. In fact, after that, shortly after 1986, based on all of the hoopla surrounding the Nobel Prize, much like the reason why the Gairdner Prize is coming to the University of Toronto. It's really to get you excited by science. It's really to give you an idea of what science is all about. People got excited by neurotrophic factors. They started looking at other possible factors that might keep neurons alive as well. In fact, we now know that there is a whole family of neurotrophins. Nerve growth factor is just one member of that family, and we know that these neurotrophins as a whole, including NGF, are actually synthesized as precursor molecules, larger precursor molecules. They are then cleaved almost in half, and that's the active molecule itself. So they are roughly 250 amino acids long, and then after some processing, they get cut basically in half to about 100 to 120 amino acids in length, and this is the active factor. So there's a larger molecule, often called a proneurotrophin, that gets cleaved into the active neurotrophin. NGF was the prototypical, the beginning, the model for all of the other growth factors that came after it. It maintains cell survival. It has to, as a nerve growth factor, it has to maintain cell survival. It causes differentiation, and it causes neurite outgrowth from that in vitro assay. Strangely, because of where the ciliary ganglion sits, this is actually not considered part of the CNS. If you were to dump on NGF onto neurons within your brain after damage, it actually doesn't work. It's not required in the CNS. We don't use it a lot within the CNS. However, another factor that was found as part of this family of neurotrophins, which is called BDNF, the one that we've been talking about with exercise and in other parts of the course related to depression, etc. BDNF is the second member of this family to have been identified. It has roughly 50% homology with NGF, so it kind of looks like a cousin to NGF. It was originally purified from pig brains. Any idea why you would use pig brains to do this? Elephant brains weren't around? Yes? So somewhat similar to the human brain. There are large quantities of it. The brain itself is much bigger, so you can get bigger quantities of neuronal tissue. There are a lot of reasons for using pig brains, and BDNF was the second factor. Soon afterwards, instead of purifying things from brain, so people would literally walk down the street to the butcher, get the brains, go back to the lab, grind up the brains, run them on different centrifuges, fractionate different parts of the brain, and try to figure out if there was any growth factors within them using very similar survival assays or growth neurite outgrowth assays. But the next family members, NT3 and NT4/5 or NT45, were actually not purified from the brain directly. What happened was they used PCR-based cloning. So once you know the sequence of a particular gene of interest that will then form a protein of interest, you can actually create degenerate primers. And these degenerate primers, you can then use that to pull out other sequences. And by doing PCR-based cloning, they found two other neurotrophic factors, NT3 and NT4/5. So of course, this was just the beginning of the entire story because, of course, these have to work on receptors. These don't work by themselves magically. And NGF binds to what is now known as the TrkA receptor. So this TrkA receptor is specific for NGF. BDNF, the second identified family member, BDNF will bind to the TrkB receptor. This is the one that it normally binds to, but other factors will also bind to the TrkB receptor. We know that NT45, the last family member to be cloned, will also bind to the TrkB receptor. Not quite as at high affinity, but it will also bind to the TrkB receptor. So will NT3. NT3 will also bind the TrkB receptor, and NT3 will bind with higher affinity to its own receptor, the TrkC receptor. All of these, though, and this is the bullet point that is shown down here at the bottom, BDNF, NGF, NT3, NT4/5, all of these bind to these receptors that I'm showing you here, but all of them will also bind to what is known as the low affinity P75 receptor. And this P75 low affinity neurotrophic receptor, or the P75 LNTR, this receptor actually doesn't look like these receptors up here at all. This receptor actually looks more like the TNF alpha receptor or the TNF receptor. And that actually is somewhat interesting. If we were to take a look at this particular receptor, this is the low affinity nerve growth factor receptor, or the P75 low affinity neurotrophic receptor. If we were to take a look, this is very, very similar to the TNF receptor. This is the overall structure. There are a number of cysteine-rich domains in the N-terminal region, and there are these card domains, or these other card domains, or cysteine-rich domains here in the N-terminal region. And that's important, but the more important thing is this domain in here. So remember, all of the different neurotrophins, the NGF, BDNF, or family members of BDNF, NT3, NT4/5, will all bind to this receptor. This receptor also contains what is known as a death domain. This is what makes up the TNF receptor. So this death domain, this type 2 death domain, is being found in a receptor that is binding to a neurotrophin. Everything that I've told you is that neurotrophins are good. Neurotrophin binding causes cell survival. It causes differentiation. It causes these things to outgrow their neurons. Then why does it also bind to this low affinity receptor, which is this TNF-like receptor that has these death domains? It was a big mystery for quite a long period of time. People were finding that it binds. People find that this exists within the nervous system. So it's not an aberrant sort of finding. And we know now that BDNF, as I showed you in the previous diagram, generally is considered to be a really good factor. And BDNF, when it binds to the TrkB receptor, will actually cause cell survival. But another form of this, pro-BDNF, the longer version, the one that doesn't get processed and becomes the shorter 100 to 120 amino acid version, it will actually bind to this P75 low affinity receptor, and it will actually cause apoptosis. So why does it do that? And why do we have these types of death domains that involve binding of these other factors? So I'm going to come back and talk to you about that next week, but I want you to keep this in mind that there are two different elements to this particular story. How many of you believe that you should memorize this diagram? How many of you believe that you shouldn't memorize this diagram? Okay, so it's somewhat Yes and no. So there are parts of it that I've explained to you that you should make sure that you know, but other things that I haven't explained to you, you don't need to memorize things that I haven't explained. So for example, this diagram, will I expect you to know this diagram? So you don't have to know this diagram. It's just to give you a different sort of look on this, that we actually have various splice variants of all of these different receptors. So the TrkA receptor, for example, has some variations. So you might be in the N-terminal domain, or it might be in the intracellular domain where it has the kinase domain. TrkB has many, many different variants of it. Some are truncated. They actually don't even have the entire kinase domain at all. So I don't need you to memorize this. I don't need you to memorize how many of these splice variants exist for TrkB or for TrkC. I want you to have an idea that we don't always have to have a kinase domain, because we don't. So for TrkB, for example, here, these two stop after the transmembrane domains. So not all neurotrophin binding to its receptor is always going to be beneficial. It depends on what receptor it's binding. So if it's binding to the P75, or if it's binding to a splice variant that doesn't have the kinase domain, it actually may not work the way that we think. And I've previously talked to you about this before, that when neurotrophin binds, and I'm going to show you the next slide, we've already looked at this slide previously, that when a neurotrophin binds to its receptor, it actually produces the kinase domain to become activated. So when we have the TrkB receptor, one subunit of it, and the identical other TrkB subunit binding to BDNF, it forms a dimer. The kinase domains become very, very close together, and the kinase domains, when they're close together, being brought together by BDNF, actually cause the kinase domains to become autophosphorylated. And this autophosphorylation is how the receptor cascade actually gets activated, and any of these following pathways that I already talked to you about then become activated. I'm hoping that you remember this diagram. So I've shown you this diagram in the last lecture, and it's just to re-emphasize this point now, that this dimerization process of the TrkA or TrkB or TrkC receptors, and now this might make a little bit more sense, that autophosphorylation that you see here as the kinase domains come together, these phosphorylation events that you see here on both sides, actually activate all three of these different pathways, ultimately leading to the things that we're talking about now, whether it's cell survival, whether it's differentiation, whether it's neurite outgrowth or neuritogenesis. All of these things are now sort of related to what we talked about before. Do you have any questions on any of this before I move on to sort of the next segment related to BDNF? Any questions? Things that you're not sure of? Yep. Sorry, KD. Have you taken any pharmacology courses previously? Biochemistry courses. So you know there's a dissociation constant, right? So how tightly something sticks to its receptors, the dissociation constant. So the more tightly it sticks, the better the KD values are, or the lower the KD values are. So that's what we look at. So some bind with really high affinity and some bind with really low affinity. Any other questions that I can answer for you? No? You guys all look scared. Are you scared? No. Don't be scared by BDNF, although this slide is somewhat scary. So I'm going to come back to this slide several times in the next 2 weeks or so. And this is, not everything on this slide is something that you need to memorize. The next few slides are actually kind of a gestalt or an overview of a lot of different things. BDNF is a really important factor. That's what the emphasis of today's lecture, and that's really what I want to get across to you. Neurons themselves produce BDNF. Neurons have the innate capability of producing their own BDNF, and this neuron here can make BDNF and secrete BDNF. This neuron over here can respond to that BDNF that's been produced, and it can also release BDNF. So there's a very complex interplay. Neurons themselves make, release, and then also need BDNF themselves. There's a very complex sort of pathway that's involved. We actually know that there are a series of steps that we can follow and are biochemically described Ultimately, what we want in our brains, the things that are important after exercise that keep neurons nice and healthy, that increase synaptic plasticity, is this factor here, mature BDNF. I told you earlier that all neurotrophic factors are produced as a pro-molecule. So we get a larger 250 amino acid molecule that subsequently gets cleaved in half that forms the mature version. So this would represent the pro-BDNF, and it's outlined for you here. And sometimes neurons simply secrete out pro-BDNF. And I'll get to that again in a second. Usually, though, inside a neuron, as it has transcriptionally formed the mRNA and it's gone through processing, this BDNF will then be sent through a series of regulated secretory vesicles where an enzyme called proconvertase will actually cleave off that pro domain that you see here, ultimately leading in BDNF release. So this proconvertase pathway is a really important one. It's found in this regulatory secretory vesicle pathway. If you're going to release BDNF, you need to have this proconvertase pathway active. If you want LTP and you want to have good vesicle release of BDNF, this proconvertase pathway is really important. So this again gets back into, we have these different variants. Neurons have a number of different receptor types. So this low affinity neurotrophic factor, for example, receptor, pro-BDNF can actually bind to this. And if it binds to this, does this have a death domain or not? So this one has a death domain. This wouldn't be beneficial to the cell. And you think, well, okay, then binding is automatically going to cause this neuron that has this receptor, because all neurons have them, to start dying. But the fact is, the neuron itself has a way to regulate this. So even if pro-BDNF binds to its low affinity nerve growth factor receptor, it will be internalized, as you see here. And once it's internalized inside the other neuron that you see here, this neuron inside of its lysosomes will actually cleave off the pro-domain so that even with this binding, if it's done quickly enough, there is no death activation, there is no apoptosis, and this neuron can then use or cause the secretion of the mature version of BDNF that you see down here. Another pathway, and I'm not going to go through all of them, the third pathway that I want to outline for you as well. There is a lot of pro-BDNF that's being secreted outside of the cell, so this pathway is actually a fairly important one. And you think that that's a bad thing. But the other positive aspect is, in the extracellular matrix surrounding the neurons, we actually have this other protease which is known as plasmin. And plasmin will interact with pro-BDNF, cleave it, and will form this mature version of the BDNF. So even though we make pro-BDNF, even though there are receptors for it, the neurons can compensate by having other mechanisms that allow BDNF, the pro-BDNF, its effects to be muted or to be blunted. And I think I'm just going to go through this one slide and then we'll take a break because there's quite a lot to go through here as well. And again, you don't have to worry about all of the different parts on this diagram. I'm not going to ask you to know the molecule known as sortilin and other things. Again, the most important parts on this diagram that I want you to make sure that you know, we have these different vesicles that you see here. So we have this pro-BDNF being coming off of the ER, the Golgi, then we form these vesicles. These vesicles are packed with either BDNF or pro-BDNF, and this pro-BDNF actually gets targeted toward the axons. We need to be able to release BDNF in a regulated way. I'm going to show you why that's important a little bit later. And we need to be able to make sure that this is all going to be packaged appropriately into these vesicles. And that's the only thing that I want to show you from this, that it gets packaged into vesicles, and the trafficking of this BDNF in these vesicles is actually a really important step. And I think because we've just been talking a lot about many different things, I think now is actually a good time to take a quick break. So Max, I'm going to need your help a little bit because we'll take our movement break now. If you want to take a quick stretch or go out and try to get some food if there's any left, please do so now while we're trying to set up for the movement break. Give us about 2 minutes. thought I'd get into something a little bit more comfortable. Hope you don't mind. There are no pockets on this thing, so I'm going to have to carry this around, but hopefully I'll be able to take you through a couple of other aspects of this. So on this particular diagram, I want you to be aware that we have a number of different places on the neuron which are actually really important. For the most part, when we're looking at how we target the different neurotrophic factors down toward the target area, the terminal, to eventually be released from these different vesicles, we had this slow axonal transport, which is on the order of about 2 to 20 millimeters per hour. It's not very fast versus the retrograde forms of transport, which tend to be much, much faster. So we know that there are fast pathways, slow pathways. BDNF takes a very slow pathway to actually work its way down toward the axon terminal. And we know that here, it can also be released from these different secretory vesicles. And that's really one thing that I want you to get from this particular diagram. The other thing that's important is that BDNF itself, working through its receptors, can actually cause CREB, or cyclic response element B, to influence gene expression. And it can, again, cause a number of different genes to be expressed and that will eventually cause cell survival. So all of these factors are important. You don't need to memorize all of the different details on this particular slide, but I did want to point these out for you. Another pathway that we've sort of looked at, which is related, and again, don't worry about all of the different details. The main point is actually underscored for you here. If this is one of the different types of TRAC receptors, that where the neurotrophin ends up can actually be incredibly important. So we know, for example, that if NGF were to bind to its receptor, like the TRAC-A receptor, that it would be endocytosed. And after being endocytosed, if NGF is binding, it can actually also undergo vesicular transport. It could cause cell body signaling, and this might cause survival as well as differentiation. That's all at the cell body. If, on the other hand, this was much farther away, this is just a cartoon figure for you, but if this was much farther away and this was at the axon terminal of a growing and developing neuron, and it happened to encounter its particular trophic factor, like NT3, it stays on the cell surface at the end of the axon. So at the cell body, it gets endocytosed. That's shown to you on the left. As it gets endocytosed, it may actually cause cell survival as well as differentiation. But as it moves, if we look on the right, if we're looking at the very end on a developing neuron as it's putting out its axon, and at the terminal end of the axon, if we had these different TRAC receptors on the cell surface, these don't get internalized at the level of the processes in the developing axon. And so the NT3 signaling here stays on the surface, and this causes local axonal outgrowth. So when you see those pictures of these filopodia and these long finger-like extensions, that's being caused by this pathway here on the right. On the left, it gets endocytosed, and it causes all of these genetic transcription changes, etc., or gene expression changes that I'm showing you here on the right. On the left. So again, make sure that you are aware that the signaling mechanisms and where they're found, whether on the cell body on the left or at the axon terminals on the right, also is a different way in which these neurotrophins add some complexity. There are many, many different complexities related to these neurotrophins. Can you think of a reason why, a real practical translational reason why people were interested in these different factors like BDNF or NGF or NT3, NT4, 5? Why might you have been interested? Why was my lab back in the late 1990s and into the early 2000s, why were we interested in finding out all these mechanisms? Ideas? They're there and designed to keep cells alive, right? Could you imagine a neurodegenerative condition where you know neurons are dying and you want to keep them alive? Maybe these neurotrophic factors might work to keep them alive. In the 1990s and the early 2000s, that was our hope. If you wanted to replace dying neurons now, how would you do so in 2017? Yep. You would probably go to stem cells. So once in a while we get these paradigm shifts where everyone starts looking at, oh, these growth factors. Neurons die in Parkinson's disease. Neurons die in Alzheimer's disease. Let's infuse these neurotrophic factors to keep them alive. That was our thought in the 1990s and early 2000s. Everyone was pursuing that, but we quickly learned that that fails. It doesn't work. And in fact, it actually becomes somewhat deleterious. And people rapidly started moving away from that hypothesis. So this is a really busy slide that I don't need you to memorize. It's really here to show you and emphasize a couple of things that I've already mentioned. So don't go crazy trying to memorize all of this, or don't get too anxious about trying to memorize all of this. This is BDNF, which we know gets into secretory vesicles. It can get released at the synaptic terminal. Now we're looking at the axon, synaptic terminal, release of BDNF, interacting with its receptor, the TrkB receptor, and causing all sorts of changes here. One of the reasons why people are still interested in BDNF, now in exercise, BDNF, and synaptic plasticity, is because this pathway has not changed over the last 20 years or so. We know that this regulated release of BDNF from the synaptic terminals, just as I outlined for you before, they get into these secretory vesicles, they get trafficked all the way down to the axon, slow axonal transport, then they get stuck here, but then they can be released almost like a neurotransmitter does when there's activity. We're interested in BDNF now because of exercise, but also because of synaptic plasticity. Because this BDNF, once it starts acting, will actually change some of the properties of this synapse that you see here on the right. And this is one of the reasons why we're particularly interested in it. And now we're interested in BDNF for completely different reasons than we started off with back in the late 90s and early 2000s. And we now know that BDNF, if you have a lot of BDNF, does everything that has been promised. Everything that we talk about with exercise, better plasticity, better responsive neurons, overall health of neurons, all of that is true. All of that mediated probably by BDNF. Does it work on, for example, if we have steady state levels of BDNF, if we were to add small quantities of BDNF, pulse on small quantities like 25 mils per hour versus having this baseline value of BDNF, we know that we have to have large amounts that are released from the synapses to get an increase in the responsiveness of that neuron. So we need more BDNF. You only get that extra BDNF that you see in this blue line at the top when you actually have activity-dependent release, when you have neurons firing and releasing BDNF. This slow, steady release, the kind of dribs and drabs of BDNF that normally get released, isn't enough to cause neurons to become potentiated or that synapse to become stronger. Again, we know that BDNF will actually enhance this factor which we call LTP. LTP is stronger in the presence of BDNF. The synapses themselves look different when we have BDNF as well. And overall, if we were to take a look at BDNF and its effects, that with acute pulses of BDNF, we will actually start to develop these really large spines that represent the postsynaptic side of the synapse as well. So we get these structural changes. If we get gradual release, we actually see that there are these longer extensions. But overall, both of these show that in terms of either having larger spines or having these filopodial-like processes, that BDNF enhances both. We get enhancement of both filopodia as well as these larger spines in the presence of BDNF. So BDNF is indeed a really good factor. And we, as I just summarized for you, if we know that neurons both can synthesize and release BDNF, and we need to have lots of BDNF being released to make a synapse stronger, there has to be a general mechanism for this to occur. So if we have a very weak sort of interaction between this synapse, this presynaptic element here at the top, and the postsynaptic element here, versus one where we get a lot of activity. So this isn't supposed to represent the size of the action potentials, but a change in the overall level of activity. If we get more activity here on the bottom, we will actually cause better release of BDNF. Better releases of BDNF will cause a better synaptic response. So this synapse actually becomes stronger. It will also grow bigger, as I've shown you in the previous slide. And there's a retrograde signaling molecule, which is known as nNOS, which completes this cycle as a positive feedback loop. So this synapse will continually signal back to release even more BDNF to make sure that this big neuron, this big synapse stays bigger. So this is a pretty good story, right? One of the things that I want you to think about is that we're interested in this. By the early 2000s, people are starting to rethink what we know about BDNF. It's not going to save neurons. Those trials in the late 1990s ended up disastrously. If you go back to those papers, our original thoughts and hopes for BDNF saving Parkinsonian patients or reversing Alzheimer's disease or being able to cure spinal cord injury, all of that didn't work. None of that worked. And it took about 5 to 7 years to realize that this wasn't going to work anywhere. But this idea that LTP, that healthy neurons need BDNF, that became the dominant theory. That's a theory that still resonates with your class today. That's what we're interested in. But I'm going to try to change your ideas about BDNF just a little bit. So one of the things that we know is that if we were to take a look across the genome, because what happened in the year 2000? So this is one of the watershed moments in biology in general. What happened in the year 2000? I'm sure you were all born in the year 2000, right? You guys are like a 98, 97-ish. So you guys were 2 or 3 years old. So you'll have known. Yes. William? The Human Genome Project. So people started looking at BDNF, the gene. Let's look at BDNF. If BDNF is so important in the brain, because it is, it doesn't work to help us from degenerating neurons, but it might be really important in brain development, etc. Let's take a look across the genome once we had it sequenced, and let's see what the importance of BDNF actually is. Magically, when you start looking at the overall genomic structure and you look at the variations that exist in populations, there turned out to be a polymorphism that was like glaring. It was like the dominant polymorphism in BDNF. And this BDNF polymorphism occurred at amino acid position number 66. You either had a methionine at position 66 or you had a valine at position 66. This was actually really big news that, wow, the population consists of a polymorphism in this really important BDNF gene. And one of the things that results from this might be that there's a change in the release of BDNF. So this might cause all sorts of different changes in episodic memory. It might cause changes in anxiety, depression, etc. If people started going in and looking at the effects of BDNF in the acute situation, not in neurodegeneration anymore, but in learning, memory, and other factors, one of the things that people knew was that BDNF signaling, if you blocked it, if you had an antibody that would bind to the receptor, so the TrkB receptor, it binds it, so BDNF cannot bind to the receptor, and you look at the effects, they knew that the number of synapses would decrease, that if you were looking in mice and you were injecting in anti-TrkB receptor antibodies, that exercise-mediated increases in memory and synaptic proteins, all of these would go back to baseline levels. So even if you were exercising and you blocked the TrkB receptor, that BDNF increase that you would normally see all vanished. So there were lots of evidence to suggest BDNF is a really important molecule for learning, memory, related to exercise, might be related to anxiety and depression. So all of these factors that we've been looking at seem to coalesce onto this BDNF. It's also particularly important within the hippocampus. We get a lot of TrkB receptors, a lot of release of BDNF, particularly within the hippocampus. All of these factors were coming together. The Human Genome Project, all of these experiments to suggest that exercise, its effects on increased synaptic proteins, all disappear if you block the BDNF pathway, suggested that this was indeed a really important one. If I've done a good job and tried to make this strong link, we also know that synaptic plasticity also regulates gene expression directly itself. And a number of really high-profile papers showed that neurotrophin, synaptic plasticity, and gene activity were all related. I happen to like their last names because they rhyme with mine, so you get Lu, Pu, and then you might get Dr. Ju. So all of these are kind of working together to suggest that this is a really important molecule. I mentioned to you also this polymorphism in BDNF. So in the early part of 2000s, people wanted to use the Genome Project, their knowledge of BDNF had failed in other areas. So once one area of research fails, you kind of use what you've learned and try to adapt what you've already learned about BDNF to other areas. And people wanted to know, BDNF, important, synapse development, important, exercise and changes in BDNF, all important, might be important in depression, might be important in anxiety. And people knew that there must be some link between BDNF and the normal structure functioning of the brain. There was a really strong indication that somehow this BDNF gene, the gene itself, was not working well in people with schizophrenia. So schizophrenia is a terrible disorder, right? It's actually a very common disorder. It affects roughly 1 in 100 individuals will have some experience with schizophrenia. There are symptoms, positive symptoms, negative symptoms. This is a very complex disorder. And somehow BDNF, this gene, was implicated in playing a key role within schizophrenia. There were two different groups that came out at the same time. I'm going to take you through the results that were shown by Sean Egan's group. And there were two groups that actually looked at the polymorphisms. This single polymorphism at position 66 for BDNF. BDNF is important, no doubt about that. Is it important in schizophrenia? If you have polymorphism of methionine or polymorphism of valine, does it really affect the hippocampal functioning and therefore affect schizophrenia? So there are all sorts of cognitive issues with people with schizophrenia. I'm not talking about the delusional aspects where they hear voices or they have hallucinations beyond the auditory hallucinations. I'm talking about their inability to understand things, to appropriately understand the world around them. That's all probably mediated through the hippocampus, and it's not working in these individuals. In an issue in cell Doing this, we were able to show that NAA is a marker for neuronal health. So again, MRS is a form of magnetic resonance spectroscopy. It uses protons, and it looks at the spin of protons. And it looks at a particular proton in N-acetyl aspartate. It also looks at molecules like creatinine, and will also look at other molecules as well, all at the same time. Any molecule with a proton in it will be detected by MRS. So I know none of that makes sense yet, so just bear with me. I need you to understand this language before I actually show you some more of this data. So do you guys do spectroscopy still in chemistry now? So Chem 130 whatever? Yes, I think. So it's just like the spectroscopy labs that you had before. You get these little peaks, and these peaks are due to the relative abundance of these different molecules that you're examining. You're not injecting anything in. All of these molecules exist within the brain. If your cells are healthy, they will have lots and lots of N-acetyl aspartate, and we'll be able to detect that they have lots and lots of this N-acetyl aspartate. So this is what you do. You take a picture that looks very much like an MRI. You then focus on an area of the brain that you want to examine the health of neurons in that particular area. So you take a high-resolution picture of this area, and you are examining the different protons that are spinning across all of the different molecules in this particular area, and you get this picture that looks a lot like this. So there are many, many molecules that are there. And we're looking for a very, very characteristic peak. And this characteristic peak that you get for NAA and acetyl aspartate, the bigger or higher this peak is, the more healthy your neurons are. Do you understand that? So far, so good? We're looking at the overall size of this peak. Now, you could also argue, I'm looking at individual variation between different individuals. How do I know that my NAA just might be higher because I'm a genetic Superman, or I like wearing tiger suits in class, or whatever the reason is? How do I know that it's not just variability? So one of the other important factors is there is this doublet that you see right beside it, which is due to choline, which is an integral part of the lipid bilayer of all cells, and creatinine. And the overall ratio of creatinine to NAA, that gives us a fairly good baseline. From person to person, it's been shown that the creatinine levels stay pretty much consistent. So your creatinine levels using MRS should be pretty similar to mine, no matter what's going on. If I have damaged neurons, my creatinine level will still be pretty much the same, but my NAA levels will go down. So we can look at this level, and we can compare it to NAA and get an overall idea of the health of individual neurons in an area of the brain that we are looking at through proton-mediated MRS. So we would get a high-resolution heat map like this. We would see that in some areas we have lots and lots of NAA compared to creatinine. In other areas we would have much less than we would have in other areas. And because creatinine levels are used, this is found to be, creatinine is relatively constant in the brain no matter what's going on in the brain. This actually gives us a good reference to actually compare it to. So we compare everyone's creatinine levels. It should all be the same, but your NAA levels will be higher or lower depending on if your neurons are healthy in an area versus not being healthy. This is the technique that was used by Egan et al. In 2003. At the time, this was not just a revolutionary technique. This was a technique that was almost like optogenetics is today. Like they came out of almost like nowhere to show that this technique could be used to determine neuronal health. We still use this technique, by the way. It's been suggested as a practical application to look at brain damage. One of the things that your generation is going to have to face, whether it's in basic research or clinical research, if you go on to professional school, for whatever reasons that we're not 100% sure of today either. There has been an increase in concussion and traumatic brain injuries. We can't tell you, for example, right now, when you should actually go back to school. So having watched people with concussion injuries, it's really hard for them to focus, to read, to concentrate. There's damage to the brain. There are all sorts of injuries to the brain. And MRS has been proposed to look at whether or not certain regions of the brain are nice and healthy. And this idea that either using MRS, the NAA levels, to see if someone's brain is getting better over time, that they might use that as a marker to actually suggest now it's okay to return to school. Your brain is picking up its health. The levels of NAA are going up or they're going back down, so you actually don't go back to school. It might be a good potential indicator. Along with another technique that is now in high use, and this is actually very common across Toronto, which is known as DTI, or diffusion tensor imaging, to look at white matter tracks. This is one way in which we might be able to diagnose concussion injuries. So MRS plus DTI might diagnose concussion injuries, because you can see a really clear break in the white matter tracks in certain regions by the arrowheads here on the bottom, where you can actually see mild traumatic brain injury on the bottom panel. This is through DTI, and we can then confirm the findings with MRS. But somehow, your generation is going to have to come up with better diagnostic markers for concussions, because we have really right now no way for us to tell you when you should go back to school, if you're going to be doing okay on writing the test, or if you're going to continue to have headaches like a month after the fact or 2 months after the fact. And again, think back to like not just athletes, but individuals who've just simply fallen and hurt their heads. So these techniques are now working their way 14 years later into some different types of practical applications. So I'm going to go through this one experiment. Traditionally, students have had a really hard time with this experiment, so I'm going to try to take you through it really carefully. If you have questions, I'm going to guarantee you that you're going to see a variation of this experiment on your final exam. So it's a good idea to jot down where you're not 100% confident in any of the techniques that I'm going to show you. And people wanted to know, why the difference? So BDNF, why does this one amino acid make the difference between having nice healthy neurons, good memory, good learning? What is the importance of that one amino acid? How could it possibly affect so many different processes, not just neuronal health through MRS, also learning and memory indices that you look at, and why is this molecule so important? So people cloned these genes. They were able to isolate these genes, and they were able to transfect or deliver these genes to cell cultured neurons. And they wanted to know, like, what's making these neurons any different, especially in these cultured hippocampal neurons, because we know that this is an important gene at the level of the hippocampus. So when they did this, they thought, okay, so if it does all of these different things, if we plop in methionine 66 versions of BDNF, they will look very different than the valine version of BDNF at position 66. So they transfect, overexpress the BDNF in these neurons, and they were surprised. There's no effect. There's no change in neurite outgrowth. These neurites grow just fine. It doesn't matter what version of this that you have. They also produce the protein at exactly the same levels. So it's not a matter of proteins not being expressed. It's not a matter of neurites not growing out. They're all interacting as they should, just like BDNF should. But when they went back and looked at the overall pattern of expression, the protein is expressed. They found that the valine 66 isoform was found in the cell body and in the dendrites, but the poorer version, the bad version if you want, is the methionine 66 BDNF version, was found only in the cell body, and in particular, it was found in areas that surrounded the nucleus. So going back to your first year biology classes, what organelle is normally found around the nucleus? So the ER. So it's around the nucleus, perinuclear. It's likely still in some sort of secretion pathway. So this is methionine 66. The other thing that they found, and I'm going to take you through this experiment on how they actually were able to do this, they also found that after transfecting in either valine or methionine 66, that when they put in valine 66 BDNF, no change in the overall appearance of the neuron, proteins are expressed at the same level, they find it in the dendrites, etc. But valine 66 was found to be secreted more easily. Did I tell you earlier that BDNF is packaged into secretory vesicles and gets trafficked down the axon? So now we know that valine 66 versions of BDNF actually get secreted as you would expect, but there was very little secretion of methionine 66. Now I told you that when we package these into vesicles, that when we're looking at a synapse, the regulated release, the secretion of these vesicles is really important. It's dependent on the level of activity. So this actually was the most important finding that these different researchers had. And I'm going to take you through this experiment, and then we'll kind of call it a day for today. So here's what they did. Here's this neuron. This is sort of a stylized version of what transfection might look like. We would very rarely do this, to be quite honest. When we have a cultured group of neurons or neurons in culture, we typically don't use a microinjection technique directly into the nucleus because it's very tedious. You actually have to find the nucleus, find a neuron, inject in, hope that the cell stays alive. It's a very time-consuming technique. People do it, but we wouldn't do it this way in 2017. So you have these different versions. This is called vector 1. So this is a plasmid that contains the BDNF gene, so either methionine 66 BDNF or valine 66 BDNF, and we fuse it. It's a fusion construct that is fused with GFP. We want to see if BDNF is actually being expressed. We also have it under the control of a generalized promoter known as cytomegalovirus promoter, or CMV. So that just gives you lots of expression when you when you introduce it into the nucleus. That is vector 1. We are also, into this same neuron, introducing the second vector, which is known as vector 2, which contains one foreign gene, which is known as DS-RED2, or DS-RED. Do you know what DS-RED is? Do any of you remember DS-RED? Those of you who took CJH with me in the summer, you should probably remember DS-RED.
Yeah.
It's just a general fluorescence marker. We have isolated it from coral proteins. Coral fluoresces red, and this DS-RED is a gene that we can also deliver. It just marks the neurons in general. It's like filling the entire room or the entire neuron with a generalized marker. So we introduce both of these genes into this single neuron. Is that clear for everyone? Do you have anything that I can explain to you now about either of these different vectors? Yes?
Could you just go over the promoter again?
So this cytomegalovirus promoter is a generalized promoter that any cell, doesn't matter if it's a neuron, it could be a glial cell, it could be a fibroblast, any cell will have the machinery to cause the transcription of the downstream gene. Okay, so this is how, for example, viruses infect cells and take over the cellular machinery. They use that particular strategy, having that promoter region. So again, we can inject directly into the nucleus. This is good if you're going to do it into cultured neurons. We can also use what are known as gene gun biolistics. This is better for tissue slices or thicker sections of living tissue. Some of you will have read about gene gun biolistics because this is the next big hope for CRISPR-Cas9. No one wants to have an injection of a virus, but now you can coat gold particles and directly inject into a target tissue like muscle using biolistics, and you don't need a virus. So biolistics is now actually coming back into style. And you can also use traditional transfection techniques, and I think you do this in your third-year labs, right? You transfect in a gene in your cultured cells. You look for expression, kind of, yes? And then you look at the overall expression of different genes after. Do you know any other methods to deliver a gene into a cell? Anything? You must all know at least one. Yes? Okay, so that's a way to regulate gene expression. But how do I actually deliver a gene into a cell? Sorry? So how do I get an optogenetic construct into a cell? I could use a virus, right? So you can also infect cells by packaging this directly into a virus as well. And there are some other techniques. So make sure that you know the techniques, make sure that you understand this. Now I'm going to show you the end results of this experiment. So keep in mind that the next series of slides are all showing the same field of view, and they're all showing neurons that have been transfected with BDNF GFP as well as DS-RED. Okay? So keep this in mind in the next slide. So before I get there, A, B, C, D, E, and F are all showing exactly the same field, all of them. So even though it looks different, we're just looking at different proteins by using different filters that look at blue or look at red, etc., on the same field. So how many neurons do you actually think are in this picture? It is a trick question, by the way. So do you think this thing, this big blob, this blue blob here in the middle is a neuron? Yes, good safe bet. This is a neuron, absolutely. So this is a neuron. We're seeing this neuron in red, in blue. It's been stained because it is stained for MAP2, which is a protein that is found within the dendrites and cell body of only neurons. So this is definitely a neuron. This is stained with MAP2 antibody, and you can see it very clearly. Here's the outline of the neuron. Here are the dendritic processes. All of this is a neuron. So again, this is just MAP2 by itself. This is with all of the different proteins overlaid on top of each other in panel F. But there is actually a second neuron. We can't see it. The second neuron that you can't see, it's not being stained with MAP2, actually is not in this picture directly. So you all agree that in D, E, and F, there is a neuron. But outside of this picture, far to the left, if we're to take a look at panel A, we actually see this neuron here that we actually did the infection or the transfection to. We delivered into this neuron, and you have to imagine its cell body is sitting way off to the left. All you are seeing is its axon coming in. This is the axon of this other neuron, this invisible, magical, mysterious neuron. And you are seeing the individual axonal processes that are coming down as these little tiny dots that you see here. Okay? So this is neuron, mysterious neuron, we'll call it mysterious neuron sitting off to the left. We can't see the cell body, it's not in this picture, but we see that it's actually making synaptic contacts with this other neuron that we see in the bottom. All good on that? Kind of good on that. Remember, we have injected DS-RED. It fills all of the processes, including the axons of the neuron that we just injected. And what we're seeing is this is the DS-RED that's being expressed. We're looking at only the axon terminal, and here we're seeing that it makes synaptic contacts with this neuron. Clear? Yes? No? Somewhat clear. Okay, awesome. What else did we inject into that neuron? We didn't just inject DS-RED. What else did we inject? We injected BDNF that is fused to GFP. And so one of the things that you see here in the middle with these arrowheads is that BDNF GFP. Now this is weird. You have to think a little bit about this. BDNF GFP could be found in the cell body of that mysterious neuron, but we also know that it gets packaged into vesicles and can also be released. And so what we're seeing here is this green represents the BDNF. Okay, so this is the BDNF that you just saw over here. That BDNF is now being expressed here. Take a look at where you're seeing it. So here are the axon terminals. Here is the BDNF. Where is that BDNF? Is it inside and only inside the axon terminals? If it was completely within the axon terminals, it would co-localize or be found exactly where the red is. Do you see any places here where green does not exist with red and therefore we don't get yellow or orange? Because when you co-localize red and green, you get yellow or orange. Do you see any pure green?
Yes.
Yes? How do you interpret that result? Yes.
Is it being transferred to the other neuron?
So it's been secreted out, and now that secreted form of BDNF is lying on top of the cell body of this second neuron here. It's exactly what we would propose. BDNF is put into these secretory vesicles, and it is then being transported to the terminals, and is then being regulated or released from the terminals as a form of regulated activity-dependent release. With one caveat, there are two isoforms. There is a methionine 66 and a valine 66. I didn't tell you which version was in this neuron. What do you think this one is? Is it methionine or valine? So this has to be the secreted form, the valine 66. The methionine, we wouldn't actually see it leaving the axon terminal. We wouldn't actually see it leaving the cell body. So this is the secreted form. Is that clear for everyone? Kind of? I'm going to go through this again in a slightly different way. Stephanie.
Are you saying that the BDNF is coming from the other neuron that we don't see?
Yes. So we put into this neuron, this neuron here, we put in two different genes, red and green. The green is BDNF. So that's been expressed in this neuron. Now, in this diagram, you see the red. So that neuronal cell body is way over here on the side, somewhere out here. It's been forming this BDNF and it's been traveling down, and that's the green that you're seeing released. So yes, we have both the BDNF and the DS red. Anything else I can answer for you now? You're going to have to know a variation of this as a short answer question on your final exam to make sure that you understand transfection, how you would look for regulated release, etc. So this is a good template for you to make sure that you understand this. So let me take this through for you in a slightly different way. So this is what a really good cell culture would look like. So these would be neurons, and this would be a cell body, another cell body over here, etc., etc. These are the processes. This would be the primary process. These are all the dendrites, etc. The axons are really hard to see because they're much smaller than a typical dendrite. We can't actually visualize them well using phase contrast. So this is when we are looking under what is known as differential interference contrast or phase contrast optics. You get to see the cell bodies in real time. I will then take an electrode and I will impale the nucleus as I've shown you here. I will introduce DS red. DS red will then fill the entire cell body processes, etc. of that particular neuron. Okay, as you see here. But we don't put in just DS red. And then under fluorescence, this is all that we would see. None of the other cells, we wouldn't see them because we've only injected this one cell. So I hope you understand why all of those other cells that were originally in this picture, we don't see them because they don't have the protein. Okay? So now we are doing fluorescent imaging. We only see fluorescence here. But we didn't have just DS red. We also put in this BDNF GFP. So there, you would actually expect to see red and green co-localizing, and we would actually see only this cell expressing both BDNF as well as GFP. Is that okay for everyone? We don't see any of the other cells in the surrounding field because they're not being injected with this particular fluorescent protein. Now, I induce activity. So again, I don't have this on the slide, so you may want to write this down. Some of you have taken other courses with me might know this. How do I induce activity in cell culture? How do I get neurons to generate action potentials in a cultured dish? What can I add to the culture dish to increase activity? How can I depolarize a neuron, in other words? Okay, I could add ions. What ions could I add? Positive ions? Can you be a little bit more specific? Calcium or sodium? Can you name me another positive ion? Potassium. So I can actually add high levels of potassium outside the cell, and when I do, the cell depolarizes, it fires action potentials, and this one cell that is also firing and depolarizing, so you should know that high levels of potassium chloride, when you add it, will actually induce activity. This will then cause the regulated activity-dependent release of that BDNF. Remember, BDNF is packaged into vesicles. It can be released when there's synaptic activity, which is now being released in these little packets that we saw earlier. Is that okay for everyone? If I were to now go back and look at all of these other cells, the ones that I didn't do this injection on, I would actually see that that one cell in the middle of that field would have all of these little green dots surrounding it. And I would stain for these neurons using an antibody against MAP2. So this is how this particular image was actually derived. So I did all of those series of experiments, and when I see these little clusters of green here, it's because I've caused the release of BDNF, and that BDNF is sitting right on top of the cell body of that particular neuron that I just stained for you earlier in this particular panel. Any questions on that? Anything that you're not clear about? So again, keeping in mind, the most important point being, not all forms of BDNF are actually equivalent. So when you hear about exercise and the release of BDNF being really important, all of that is true. However, it also depends a little bit on your polymorphism. If you have the valine version, exercise is much more beneficial to you than if you had the methionine version, because you just don't secrete as much. No matter how much regulated activity there is, no matter how much everything else is going on, that simply isn't going to be as beneficial. So I know we've covered a lot today, so there's a lot to digest. I know I have some other slides remaining, and I can go through them quickly at the beginning of the next lecture, but I think I'm just going to actually end that here now, and then we'll continue on in the next lecture. Do you have any other questions or anything that I can clear up for you now? If you haven't done so yet, please make sure that you submit your visual summaries. The TAs and I are going to start working them starting on Thursday evening into Friday. Some of you will start getting the grades back and some comments back on your visual summaries starting on the weekend. I'm hoping you all did a great job on it. I'm sure you all did. And again, have a great weekend. I'll see you all next week.